Nerve cells are responsible for what we see, what we smell and taste and feel. They are responsible for our memories. They are responsible for our decisions, our drives. How do these nerve cells, accomplish these tasks? Well, they do two things. One is they can transmit electrical messages along the length of the neuron. And given that some neurons can be quite long, so for example, there are individual neurons which stretch from the tip of my toe to my medulla oblongata in my brain, so they can be quite large. And when an electrical message reaches the end of a cell, this is where chemicals such as neurotransmitters can be used to transmit a message from one cell to another. This video will be about the electrical message which travels along this axon, the action potential, and the next video will be about the chemical communication which occurs at the synaptic knob. Now, while I want to focus on the action potential, there then are other topics which are relevant in the lecture after that. So for example, how does this region of the neuron decide when an action potential should be initiated? This is what is known as the trigger zone or the axon hillock. After summating all of the input from the dendrites and the soma. So that will be yet another topic. But once again, the big topic here is how is it that a cell can conduct electricity? So first point is that this cell membrane, as you may remember, has polar phospholipids heads, which face the polar watery environment of the outside of the cell and the polar watery environment of the cytosol. But between the two are these non-polar fatty acids that then form a barrier to polar molecules. Polar molecules cannot just zip in and out of a membrane, such as ions which have charge. So in these animations, sodium will be red, potassium will be blue, and chlorine, where it comes up, will be green. So these ions have a charge, positive for sodium and potassium, negative for chlorine, and charged molecules cannot pass through these nonpolar membranes. So cells can then synthesize ion channels which allow the passage of these charged particles through the membrane. So the charged particles can pass through the membrane, but only in the proteins that the cell produces. And the cell can then decide which ion channels to make, how much of the ion channels to make. And so this is under the decision of the cells themselves. Now, one of the most important proteins in the body is not a passive pump like seen in the previous videos, where ions can simply go from an area of high concentration to low through diffusion, but instead what is an active transport pump which uses ATP. This is known as the sodium-potassium ion exchange pump. And what it does is use ATP to pump sodium and ions in the opposite direction that they would want to diffuse. So here you see ATP, and its energy is going to be required. This protein is going to break the triphosphate bond here and use the potential energy stored here to pump ions against their concentration gradient. So sodium is most highly concentrated outside the cell. The few sodiums which are inside the cell, they would prefer to stay inside the cell, just acting on their diffusion gradient, and they would prefer not to go outside. So these sodium ions going outside would be acting against their concentration gradient, going from an area of low concentration inside the cell to an area of higher concentration outside the cell. But this sodium-potassium exchange pump does take 3 of these at a time by using the energy here in the triphosphate bond. It will pump 3 sodiums out. Now, sodiums are positively charged, and 3 of them are leaving the cell. But at the same time, then, 2 potassiums, which are outside the cell, will be pumped in. Potassium, likewise, is moving against its concentration gradient. There's a lot of potassium inside the cell, not much outside the cell. So left to diffusion, potassium would prefer to go from inside of the cell to out, but here we are pumping this ion from the outside in. And then more ATP will bind and more sodium ions will get pumped out. And so this pump runs continually, pumping sodium out of the cell so that the outside of the cell has a high sodium concentration, and pumping potassium inside the cell so that inside the cell has a high potassium concentration. Now, most cells of the body use this pump, and for that reason, most of the body's potassium is inside cells, and most of the body's sodium is in extracellular fluids like blood, plasma, lymph, etc. And so both of these are positively charged ions. Potassium is positive, sodium is positive. But because we pump 3 positives out and bring 2 positives in, 3 positive sodiums leave, 2 positive potassiums come in. As a result, more positive charge leaves than comes in, and thus the outside of the cell has a positive charge, and the inside of the cell has a negative charge. We can measure the separation of charge in what's called voltage. That's what voltage is. It measures the separation of charge. This sodium-potassium exchange pump has created voltage across the cell membrane so that there is more positive outside, more negative inside. We could measure the voltage difference across the cell membrane with millivolts. So for example, this varies from cell to cell and even among neurons. So some neurons it's a little higher, lower, some muscle a little higher or lower. But there is what is known as a resting membrane potential, which could say be minus 70 millivolts, which is a negative charge because the inside of the cell has a negative charge compared to the outside. If that was the end of the story, then we wouldn't think, we wouldn't move, because sodium would be outside, the axon, potassium would be inside, and that would be that. However, there are other ion channels which will let sodium go the direction that it wants from an area of high concentration to low. There are gates which will let potassium go the direction it wants from an area of high concentration to low. These are not passive channels which are open all the time. These are gated channels which are sometimes closed and sometimes open. So there will be certain times where electric charge is crossing the cell membrane, and then other times when it is not, based on whether these gates are open or closed. What determines whether they are open or closed? Some of these are chemically gated or ligand gated. So right now this gate is closed, but there are chemicals which can bind to the gate, like a neurotransmitter. Say the one that I'll be using in an example today is acetylcholine. So acetylcholine can bind to this channel. And when it does, this will then change the channel so that now it is open. And so whether this sodium gate is open or closed depends on chemicals. When chemicals like neurotransmitters are present, then the gate will open, and sodium can go from an area of high concentration outside the cell to a lower concentration inside the cell. However, if we were to break up this acetylcholine, as will happen, there are enzymes that do that, it gets broken into two halves, acetate and choline. And then this gate returns to its closed position. And now sodium, once again, even though the concentration gradient would drive it from the outside of the cell to the inside of the cell, will not be able to cross the membrane. So there are ion channels which are ligand gated or chemically gated. There are other channels which are voltage regulated. Once again, voltage is the measure of the electrical difference across the cell membrane. And this is determined by the concentration of the charged particles, some positive, some negative, where they are on either side of the cell membrane. And so this ion channel could let sodium pass through and allow it to go from an area of high concentration outside the cell to a lower concentration inside the cell. But right now it's closed. But what would change that is if the cell membrane were to change its voltage. If the electrical difference across the cell membrane were now different than what it is, say, at rest when this gate is closed, it would then open, and then the sodium would be permitted to pass from an area of high concentration to low. The action potential, which we'll be talking about in this video, does just that. The action potential changes the membrane voltage, which then changes these voltage regulated gates. So there are sodium gates, which are voltage regulated. At one voltage, they are open, and at another voltage, they are closed. And then in the same way, there are potassium voltage regulated gates. So remember that potassium is concentrated inside the cell and would like to leave. However, this gate is closed at the resting membrane potential. But then during the action potential, once again, it will open, and now the potassium ions are permitted then to go from an area of high concentration to low. And so the sodium-potassium exchange pump establishes a resting membrane voltage with sodium outside and potassium inside. But there are ion channels which will open under certain conditions. Some of them are chemically gated or ligand gated. So when a chemical like a neurotransmitter binds, they could open. Some of them are voltage regulated so that at a certain voltage, they would open. Now, let's talk about what that would do. All right, so if we were to measure the voltage across the cell membrane, and so once again, voltage is an electrical difference, and because 3 positive charges are pumped out for every 2 positive charges pumped in, the resting membrane potential is negative. There are more negative charges inside the cell than outside. And the difference can vary from cell to cell, but in neurons at minus 70 millivolts is a common number. So that's the resting membrane potential. And if nothing happened, then it would stay at minus 70. But if sodium ions were allowed to enter the cell for whatever reason, so there could be passive ion channels. This could be, say, a touch neuron, and you know, there could be a response to the physical touching. There could be neurotransmitters on dendrites causing gates to open. So therefore, there could be a number of stimuli which would open a channel which allows sodium to come in. What does that do? Well, sodium is positive. And so if a positive charge comes into the cell, that means this cell becomes more positive and thus less negative. So if its resting membrane potential was here at minus 70 and here was 0, we then move closer to 0. We are moving towards the positive end. And so this is what we call depolarization. So when positive charges come into the cell, that will make them less negative and thus more positive. And so this is depolarization. So if a change happened so that sodium came into the cell would depolarize. Now, if we don't uh, reach an action potential, then the sodium potassium ion exchange pumps will pump the sodium back out, and then it would return to the resting membrane potential of around -70. There are also then potassium agates. And if the potassium gate opens and potassium charges are allowed to leave, well, what that would do is the cell is already negative, and if more positive charge left the cell, that would make it more negative. All right, so when positive charge leaves a cell, the cell becomes more negative. And so if something were to happen, and this could be a response to neurotransmitters, or could be a number of things, and potassium gates open, then potassium ions would leave, and the cell would what's called hyperpolarize. So it normally has a resting membrane potential. If it becomes more negative than that, that is referred to as hyperpolarization. And so here, as potassium charges leave, the cell hyperpolarizes. Once again, that sodium-potassium exchange pump will grab potassiums outside the cell and bring them in. So eventually, its action will then return the neuron to its resting membrane potential. Okay. With all of this, then, we are ready to discuss the action potential in neurons. And once again, this is critically important. Nerve cells, neurons in general, do two things. They have electrical action potentials travel along their lengths, and then a chemical message in the form of neurotransmitters can jump from one cell to another. And thus, action potentials are a foundation for how we think, how we move, what we feel, what we remember, etc. So let's explain it. At rest, there are more sodiums outside the cell than there are positive potassiums inside the cell. So there is a resting membrane potential of -70 millivolts. Okay, and that was once again thanks to the sodium-potassium ion exchange pump. I apologize for that little sodium ion going across. That one was misbehaving, sorry. If we could get to what's called the threshold voltage, I will define that in just a second. If we get to the threshold voltage, then these sodium gates will open, and sodium will now be allowed to come rushing in. All right, and so this is a voltage regulated channel. And there's a special voltage called the threshold voltage, which will cause the voltage regulated sodium channel to open. Sodium can come rushing in. As it does so, that will change the membrane potential. It will no longer be -70. So much sodium rushes in that the membrane potential will rush up to +30 millivolts. At that point, this potassium gate, which is also voltage regulated, will open. So at +30 millivolts, the sodium gate, which was open, closes, and the potassium gate, which was closed, now opens, and now the potassium ions will be allowed to leave the cell. And this membrane voltage, which was in the positive zone at +30, then becomes negative again as all of this positive potassium ions as it then rushes out. And then the cell returns to its -70 millivolt resting potential, more or less. It'll actually dip a little bit under -70 millivolts. That's what's called an after hyperpolarization. And then afterwards, the sodium-potassium exchange pumps have to then return to taking the sodium that had come into the cell, pumping it out, and getting the potassium which had pumped out of the cell and pumping it back in. Now, these sodium-potassium exchange pumps are so important that a cell can spend, say, um, a quarter of its resting energy budget just on that. So an enormous amount of the energy that you have used over the course of your life is pumping sodium and potassium outside or on either side of cell membranes, such as in nerve cells and muscle. All right. So before we put all of this together, once again, so the sodium wants to come in, going from an area of high concentration to low, and it can't until this voltage regulated gate opens. So at one voltage called the threshold voltage, this opens, all right, and sodium comes rushing into the cell. The gate then closes at around the point where the cell has reached 30 millivolts on the positive end because of all of the sodium then coming in. And then once again, there was a voltage regulated potassium gate. The potassium wants to leave the cell, and it can't as long as this gate is closed. But when we reach minus 70 millivolts, then this voltage regulated potassium gate opens. Potassium then is allowed to leave the cell, and then does so, going from an area of high concentration to low, and then this causes the cell to repolarize, going back down into the negative zone. So putting it all together, we see this, that here the cell has a resting membrane potential at minus 70 millivolts. Now it's waiting for a stimulus. Now let's say there is a bit of a stimulus, and I'm sorry, this is the wrong video. I'm sorry, I thought I had another video here. So let's say that there is a stimulus to this neuron, and the stimulus causes sodium ions to enter somewhere in the cell, and that the resting membrane goes from minus 70 millivolts to minus 65. What happens? Nothing. Nothing happens, and eventually the sodium-potassium exchange pump will pump those sodium ions back out. So I'm going from minus 75 to minus 65. As some sodium ions come in, that doesn't accomplish anything, and it doesn't get to an action potential. If more sodium ions come in, going from minus 70 to minus 60, what happens? Nothing. The sodium ions will be pumped out. What happens if enough sodium ions come into the cell to get to minus 55 millivolts? Everything. So this threshold voltage of minus 55 millivolts is sometimes described in what is known as the all or none phenomena. If you cross the threshold voltage, you get an action potential. If you don't cross the threshold voltage, you don't get an action potential. So you don't get halfway and get a little action potential, or etc. You either get an action potential or you don't. It's an all or none phenomena. And that is because at minus 55 millivolts, that is the voltage where these sodium gates, which are voltage regulated, they open. They open, which allows sodium to come into the cell, and look at what happens. So at minus 55 millivolts, the voltage regulated sodium gates come into the cell, and so much positive charge comes into the cell that the membrane potential continues to rise, even going into the positive realm of around plus 30 millivolts. At that point, the sodium gates will close, all right? So at minus 55 millivolts, the sodium gates open and the action potential begins. Sodium comes rushing in, the cell depolarizes, and at plus 30 millivolts, the sodium gates close. But then these are voltage regulated as well. Now, why do we want them to open? Well, I apologize, usually I act this out in a class. But like if you're trying to run away from something, all right, the goal is not to have your muscles, do this in mid-pace. It's that you tell your muscles to contract and then you stop. All right, and then so you can take another step and tell them again, and take another step and tell them again, and take another step and tell them again. So it's not like we're going to throw away this neuron after one usage, okay? We need it to get back to the resting membrane potential, right? Because we don't do this and now we're done with our hand. I mean, if you wanted to do this, you have to tell the muscle to contract and then stop telling it to contract, so that you can then do it again. And so here we are at plus 30 millivolts, but that doesn't work. We're going to have to get it back down to the resting membrane potential. That means that means positive charge is going to have to leave. Now, sodium is a positive ion, but it just came in going from an area of high concentration to low. We could ask it to leave nicely, but it's not going to. But potassium is dying to leave. Potassium wants to exit the cell because there's such a high concentration of potassium ions here. And at plus 30 millivolts, these potassium ion gates open. And now these potassium gates are allowed to go the way that they would like to, leaving the cell. And notice what's happening to the membrane potential. It's dropping down. This is what's known as repolarization. All right, so it's going back into the negative realm, and it even then makes a little extra dip called after hyperpolarization. So there's hyperpolarization there. Once we get back to this realm of the resting membrane potential, then as I had said, the sodium-potassium exchange pump can then take the sodium which had come in, pump it out, take the potassium which had left, and then pump it back in. So I think a lot of students at this point, this has been a little more, voltage discussions than they're comfortable with. And now that we've done all of this work and the neuron is exactly where it was before. So it started at minus 70, it goes up to plus 30 millivolts, it comes back down to minus 70 millivolts. What have we accomplished? Well, if you're just looking at one part of the axon, nothing. We're back to where we started. Now, here's where you see it makes all the difference. And just for clarity's sake, very often what I do at the beginning of this lecture is I ask my students to turn to someone near them and hit them. Okay, and so some do it right away, but some take some convincing. But the question then is, how did you send that message? So how did a message, go from this brain to your arm muscles that you hit this? How did it spread over the arm muscle? And then when someone hit you, right, and I ask, well, what do you do? It's like, oh, you hit them back. And so how did you decide that? These are messages which have to travel. Remember that cells are microscopic, but here we're going from here to here to here, that's a foot, 2 feet. All right, so how are cells transmitting messages over feet? Well, here's how. Because as this area depolarizes to threshold, sodium comes rushing in. Now, yes, sodium comes in, the membrane potential goes up to plus 30, then the potassium gates open, and then it goes back down to minus 70. And this part of the membrane then returns to normal. I apologize that zooming and watching animations sometimes doesn't go well together, so it's glitching and flows more freely otherwise. But the point was that the sodium that came rushing in here caused this area to go from minus 70 to minus 65 to minus 55. This area then crossed the threshold voltage. That meant that here sodium came rushing in. Now, it's true that sodium will come rushing in. The membrane potential goes up to plus 30 millivolts. Then the potassium gates will open and the area will go back down to minus 70 millivolts. But while that's happening, the sodium that came in, it now goes to this area, causing this area to depolarize to threshold. So sodium comes rushing in. And that will cause this area to depolarize to threshold, and sodium comes rushing in. And so what you see is this wave of charged particles crossing the cell membrane. And we call that electricity, all right? When you have a movement of charged particles, all right, changing the voltage, this is an electrical message which is now traveling along the length of an axon. And if you hit someone and you ask, how did a message go from here to to my arm muscles, etc.? Well, through the action potentials, because even though the soma, which decides, hey, you need to send a message to that muscle, is up here, it has an axon which goes through the brainstem, through the corticospinal tract, and then after a synapse, it then goes out to the arm muscles. So even though all of the changes seen here, that's what's happening at this spot of the axon. What happens at this spot then causes an action potential at the next spot, which causes an action potential at the next spot and the next spot. And so thus a message can spread along the length of the axon. Okay. Last point is how fast. All right, well, this can vary. And so this could be traveling, say, at 2 miles an hour. And say during a class, sometimes I say, imagine I'm leaning up on a hot stove. This message that I touched a hot stove is now traveling at 2 miles an hour towards my central nervous system, and the message then to pull my arm away travels at 2 miles an hour in return. But that might not be fast enough. The longer my hand stays on the hot stove, the more tissue damage that I incur. And so there are ways that the message could be speeded up. For example, axons can more easily allow the movement of particles if they are wider. So a wide axon will transmit a message more rapidly than a narrow axon. And then finally, since the electrical message is charged, okay, so this is the movement of charged particles, some things inhibit that, such as lipids. And if you recall, there are glial cells, oligodendrocytes in the central nervous system, Schwann cells in the peripheral nervous system. There are glial cells, which then make a lipid protein mix called myelin, which then gets wrapped around the axon. So here's a glial cell, which has put myelin at this spot. Here's a glial cell, which has put a myelin at this spot, etc. So each of these is a cell. Now, the places where they meet, we refer to those as nodes. So this is a node, and the area where the lipid is, that is the internode. So most axons then have the electrical message go over each and every section of the axon. But since charged particles are not, electricity is not conducted by lipids, here the charge is jumping from node to node. That's what's called saltatory conduction. So if you speak Spanish, saltar is to jump. So the action potential jumps from node to node to node to node, and then travels much faster. So if we were to compare 3 axons, one thin and unmyelinated, one thick and unmyelinated, and one thick and myelinated, they would then conduct an action potential at different speeds. The axon which was thin and unmyelinated, that would take the longest. The axon which was thick and myelinated, that would then have the most rapid speed of transmission, maybe 280 miles per hour. So over 100 times faster than the thin unmyelinated one. Well, you might say, oh, all right, so once again, so here you can see the most rapid transmission is on the axon which is thick and myelinated. A thick unmyelinated axon then transmits the message faster than a thin unmyelinated axon. Well, this is great. And you could ask the question, well then, speed is good. Why aren't all of my axons thick and myelinated. And there are two reasons for that. One is just a space issue. If every axon that you had was, say, thick and myelinated, then the nerves that I have in my arm would be as thick as garden hoses, and my spinal cord would be the size of a garbage can. There's no room for that, obviously. There's a space issue. But then secondly, not all messages are urgent. So if I want, to sweat, do I have to tell my sweat glands, please start sweating now? Do I have to tell, hey, I'd like you to move the breakfast that I ate, a little farther along the jejunum of my small intestine? Does that message have to get there now? No, it doesn't. It's not urgent. You can wait another split second for that message to get there. And so while we have ways of speeding messages up, not all messages are urgent, and it would change how much space we need for our nervous system if every neuron was wide and myelinated. So the nerve cells, which are responsible for thought and drives and memories and movement, etc., they're doing two things primarily. They're conducting electrical messages along their length in what's known as an action potential. And then where cells touch each other, there's typically chemical communication in what is known as a synaptic chemical communication. This video was about the electrical action potential. The next one will be about the chemical communication at synapses.